Hello, everyone, and welcome. Thank you for joining us today for the first webinar in our new series titled The New World Screwworm, Past Lessons, Present Programs, Future Frontiers. My name is Justin Overcash, and I'm with the Foundation for the National Institutes of Health, where I work as part of the Gene Convene Global Collaborative. Gene Convene's mission is to support and advance best practices and informed decision-making for the development and governance of genetic and biological control technologies to improve public and animal health. We do this by convening diverse expertise, identifying key scientific and policy questions, and supporting transparent information sharing and capacity building across sectors. This webinar series focuses on the New World Screwworm, a pest that has historically caused devastating impacts on livestock, wildlife, and human health, and whose successful control stands as one of the most significant achievements in area-wide pest management. At the same time, recent detections and northward spread underscore that it is not just a historical story, but remains a contemporary and forward-looking challenge. Across this series, we will examine past lessons from eradication efforts and international coordination, present programs supporting surveillance, preparedness, and containment, and future frontiers, including new scientific tools, technologies, and policy approaches that may strengthen long-term control and prevention. Before we get started, I'll cover a few housekeeping reminders. If you experience any technical issues during the webinar, please email events@fnih.org. Your video and audio will be off during the session. Today's presentation will run approximately 45 to 60 minutes, but please feel free to submit questions at any time using the chat function. We will address questions during the discussion period at the end of the presentation as time allows. The webinar is being recorded, and recordings will be posted on the Gene Convene Virtual Institute, as well as the Gene Convene Virtual Institute YouTube channel following the event. Today's webinar is titled The New World Screwworm, a 50-Year Success and a Renewed Challenge, and we're pleased to be joined by Dr. Phillip Kaufman. Dr. Kaufman is a professor and the head of the Department of Entomology at Texas A&M University. Dr. Kaufman earned his BS in Animal Science from the University of Illinois, his MS in Entomology from the University of Wisconsin-Madison, and his PhD in Entomology from the University of Wyoming. Over the course of his career, he has held faculty appointments at Cornell University and the University of Florida, and he brings decades of experience spanning research, teaching, extension services, outreach, and administration. Dr. Kaufman's research program focuses on the biology, ecology, and management of arthropod pests of veterinary, agricultural, and public health importance, with a particular emphasis on developing innovative control tools in the face of increasing insecticide resistance. His work has contributed to new pest management strategies across poultry, bovine, equine, and companion animals. And he has authored more than 130 peer-reviewed publications. Particularly significant for today's discussion, Dr. Kaufman currently serves as the chair of the Texas A&M AgriLife New World Screwworm Task Force, where he helps to coordinate the research and extension response as this destructive pest once again threatens livestock in Texas. Before we begin, a quick reminder that I will be moderating the Q&A session following the presentation, but of course, please post your questions in the chat at any point during the talk. As mentioned earlier, this presentation is being recorded, and with that, I'll turn it over to our speaker, Dr. Kaufman. Whenever you're ready, please go ahead and share your slides.
Thank you for having me on today. I'm more than happy to share this information, try and bring everyone up to speed so you're better informed with this challenge that is a livestock entomologist. I never thought I would ever be working on this fly, and I find myself deep in the trenches with my colleagues trying to prepare the U.S. for a potential return of the screwworm and developing new approaches to help re-eradicate this fly. And so Justin covered a lot of this background on me. So I am leading the task force for the screwworm response with our AgriLife, which is our research and extension components of Texas A&M University, our college, working with the College of Veterinary Medicine, other department heads to coordinate our efforts so that we're not duplicative and we identify the needs of the various groups in Texas for what they are asking for and coming up with publications and other support for them. I also am a participant each week with what's called the Texas Coalition for Screwworm, which is a collection of all the commodity groups in Texas that include livestock as well as the wildlife groups. Because if you're not familiar, Texas has a very active hunting industry that in many cases exceeds the value of our agricultural commodities in the livestock area. Um, in Texas, Texas Animal Health Commission and Texas Parks and Wildlife Department are considered by our governor to be the lead state agencies in developing a response at the statewide level. And I've worked with those individuals developing what will be the response plan should the fly arrive. And worked in a number of other capacities, including educating our legislative groups, both at the state and federal levels, and uh, serving as a liaison to USDA Agricultural Research Service and APHIS, both of whom are tasked with part of this response to New World Screwworm. So you go through some background of the biology of this pest, and coming at this as if not expecting anyone to have taken an entomology course. So this is the New World Screwworm adult fly, Cochliomyia hominivorax. The larvae of this fly are obligate parasites, which means they can only survive uh, in living warm-blooded animals in this case, which means our livestock, our wildlife, our companion animals, ourselves, and it has been reported in birds, but it's fairly rare to occur in birds. And when we have living maggots in a living organism, such as a mammal, that term is called myiasis. If we look at the scientific name, hominivorax, the specific part of the name, essentially translates from Latin to be man-eater, because this fly certainly does attack humans, which makes it a public health risk. And when you have an infestation of the larvae on an animal, this can and often does lead to fatalities in the infested host. And that's what brings its importance, besides just simply eating our tissue, that it actually causes death is quite problematic. So the New World screwworm is not a normal fly. It has what I've seen in giving talks is a lot of ranchers think that it's a fly that sits on the animal all the time, like a horn fly or a stable fly. It most certainly doesn't. And early in the education program, we had created a website where people could send us pictures if they thought they saw one. And what we were learning from that is that we needed to explain to people that it was exceptionally rare for someone to see an adult screwworm unless you are actively baiting for the fly in an area where screwworms existed. This fly has no interest in manure, which a lot of our normal flies do, and it does not enter barns the way our other flies do. It also does not stay on the animals. That female fly will come to an animal to lay her eggs and then she leaves. They spend most of their time in shaded habitats, so wooded areas, um, where they're finding microclimates that are suitable for them, and then they visit those animals in pastures or fields that are nearby when they're searching for a host to oviposit on. It's been reported that the flies will cluster in these habitats, these off-host habitats, but that's likely because there are probably few areas that have the microclimates that the flies really are interested in, and the flies are congregating to the microclimate, not necessarily to each other. And interestingly, New World screwworm is been reported that it does not fly over large expanses of water. And we've seen that through historical distribution of the fly in North America, where the Mississippi River was most definitely a biological barrier for the fly to expand into the southeastern U.S. And we'll explain how southeastern U.S. eventually received screwworms, but it essentially came down to humans creating situations where we were moving the insects ourselves. So this is a tropical fly that makes incursions into temperate climates. This is a native species to the Americas, and I have on the slide the historical geographic range where it says that it would make it into southern Canada. And when they talk about that, they're really talking about this area over in Ontario, just above Lake Erie. But again, these are movements of humans moving infested livestock to very northern areas and the fly establishing during the summertime. So the red line on here is essentially the 41st parallel, and that's the traditional northern expansion of the fly before we eradicated the fly in the 1960s. And again, most of the movement up toward that line were humans moving infested animals. Either we didn't know it or we didn't care back at the time. It has, again, a tropical fly, so its preferred activity is in the 65 to 91 degree Fahrenheit zone, but it really does not like cold weather at all. And so the adults become inactive below 60 degrees Fahrenheit, and prolonged exposure to those lower temperatures essentially kills the fly. And so we have these winter diebacks. And in the slide you're seeing, the very dark areas are the overwintering populations. And you can see, depending on how cold the winter was, a lot of Texas could be having year-round activity of New World screwworm. This fly, like many of our other higher flies, so non-mosquito flies, is active only during daylight hours, which is very good. So it's like when you have a picnic, you during the day you have house flies and other flies bothering you, but if you have a party at night, you have mosquitoes bothering you, and it's because of the diurnal activity of those different organisms and what their preferences are. This fly does not move around at night. All right, when we talk about screwworms, there actually are two species in the Americas. There's actually four, but there's two that you might regularly encounter. Cochliomyia hominivorax, which is the New World screwworm, and Cochliomyia macellaria, which is called the secondary screwworm. Says historically, if you look way back in the records for New World screwworm, it would have been called the primary screwworm because it was the insect that primarily started myiasis infestation. The secondary screwworm would come in secondarily to the primary screwworm or New World screwworm starting an infestation. And so the secondary screwworm really is a carrion feeding fly, like most of the rest of our blowflies. The New World screwworm, probably millions of years ago, did a host switch from carrion to feeding on living organisms, which is actually not something that you would normally expect because a life of parasitism is quite a bit more risky than life is feeding on carrion. But macellaria, the secondary screwworm, is probably the most common blowfly in Texas. You see it everywhere. If you were to take some rotting liver or rotting meat and set it out in your backyard very quickly, if it were during the summer, very quickly, you would have blowflies come in. And one of the early colonizers of that is Cochliomyia macellaria, the secondary screwworm. So differentiating these two is very important, but it does require a trained expert, which is why we have some of the conditions for identification built into the response plan. So you're looking at these going, well, obviously they're easy to tell apart. The one on the left, the New World screwworm's blue, and the secondary screwworm is green. But in insects, particularly blowflies and other metallic organisms, color is not always a reliable feature because color is essentially the reflection of light, which is the structural features on the exoskeleton. And screwworms will change color with age. And if you go into what our colleagues in Central America tell us is that most of the screwworm, the New World screwworms down there, are actually more green than blue, as depicted in this picture. So there are some other key features. I'm going to rapidly go through some on the next couple of slides, but if you look behind the eyes on a New World screwworm, often there is an orange triangle that is indicative of it being a New World screwworm, whereas on the secondary screwworm, that never is present. But that orange triangle is not always present on the New World screwworm. Uh, what we do use are the very distinctive facial hairs, and I'll try and show those on some slides here in the next couple slides. Um, and you've heard people talk about these stripes on the back of the fly being diagnostic, that they're shorter in one, longer in the other. Um, they tend to be not especially reliable as a primary indicator of which species they are. And so what we tell people is, if you're collecting flies or you think you found a screwworm, if it's metallic blue or green with stripes on the back, go ahead and turn it in and let an expert take a look at it and see if they can use some of the other characteristics to uh, identify that fly. All right, so it also depends whether they're a male or a female. So on the upper left corner, we have two females of Macellaria and hominivorax. We can tell these are females because there's a gap between the eyes on top of the head, whereas on males, the eyes nearly touch. But just looking at those insects, you probably can't tell the difference between them. But what we're going to look at, and I'll use the male here, it's a little bigger picture. We're going to look at some of the features on the hairs on the front of the face. And, but again, trained expert is required for these. So there are some characters that are only present in females and some that are only present in males. So these are females. And what we're looking for, again, you can see the orange dot or triangle behind the head on the New World screwworm. On the secondary, there's not the orange. You can look under the base of the wing and whether there are these orange hairs or setae on the secondary screwworm, but not on the New World screwworm. And the other features come down into colors of hairs that are present in here. Again, requires a microscope and someone who's trained to look for these. So on the male flies, they again often will have that orange dot or orange triangle. You're looking at these, whether in the New World screwworm, are these hairs all black or do you have some light colored hairs mixed in with them? And this is a little different picture. It's not these heavy setae, it's these very small ones. So it's a very difficult fly to identify. And that's why we have groups of people who are undergoing training, multiple levels of training, and that training, they have to very accurately tell the difference between these flies. We do have molecular techniques that can be used, but of course you can identify an insect quite a bit faster than you can run a PCR. There are a couple of features that are often present. So in the secondary screwworm, these are females. They'll have these hairs around the tip of the abdomen, whereas on the New World screwworm, they're generally not present. But that doesn't really work on the males. Larvae, excuse me, larvae tend to be a bit easier to identify in many ways, but part of that comes down to how well the larvae are preserved. So on these larvae, we have segments and they have numbers on them here. And it's going to matter when we get down to this 9th and 10th segment, what some of those features look like between the two flies. Um, insects don't have lungs. They breathe through a series of branched tracheae, which think of them as straws that branch and branch and branch. The primary openings, which are called spiracles, are on the back end. So this is the back end of the fly. The pointed end is the head. And they open to the outside, and you can see these tracheal trunks inside on some of these maggots. But there also are breathing openings up in the front, which are up here and around the second and third segments. And you can look at the way those structures are built can provide you with identification. What we really like to look at are these spines. And on these insects, there some zoomed in pictures. The spines are essentially very short hairs that are pointed, almost triangular, and they're used for locomotion, among other things. And it's part of why the fly gets the name screwworm, because if you look at it, it's pointed at one end, fat at the other, and it looks like a screw with these dark setae that you can see on the pictures at the top. And then here's the tracheal trunks that I mentioned. So the opening is back here. And on the New World screwworm, they tend to be very darkened, whereas on the secondary screwworm, they're not darkened, or we call sclerotized. And I'll show you some additional pictures of those. So how do we tell them apart? All right. So if we look at the ring of spines that are between the ninth and 10th segments, it's whether the ring of spines encircles the fly or whether there's a gap. So in the New World screwworm, the spines encircle the entire ninth to 10th segment, whereas in the secondary screwworm, there is a notable lack of spines on one side of that fly larva. And that is very distinguishing characteristic. The other, this is those darkened tracheal trunks, and we'll take those apart here in a second because they're not always present or not always obvious from the outside. So these pigmented tracheal trunks on the New World screwworm, you can sometimes see them through the larva, but sometimes you can't. And so what they'll have to do, because often we get larvae submitted that are in this shape, right? They're not beautiful pictures like we have at the top. They maybe are darkened, and that usually means that they were not put into alcohol, or the alcohol concentration wasn't strong enough to kill all the bacteria, and the inside of the maggot starts to rot as the bacteria consume it before the alcohol soaks through that larva. And so we can dissect it, you pull apart the back end of it and see if you can then identify the pigmented trunks. This is two pictures that are both New World screwworm. The one on the bottom right is a dissected secondary screwworm where those tracheal trunks really are not pigmented. Another, if you have the pigmented tracheal trunks, you have a New World screwworm. But again, all these larvae need to be submitted to the proper authorities regardless. Unless you are a certified identifier, you are required to submit any live larvae that are taken from a live animal to the NVSL or your Department of Agriculture. So very quickly, when we look at screwworm life cycle, you have the pupae are in the soil or in the ground underneath leaf litter. They're just trying, when they go to pupate, they just try and get away from the sun. After a period of time, that fly pops the cap off of the end of the puparium and crawls out. And I'll just have you note this little white thing you see at the front of the fly. That is a sac that they push out between their eyes that pops the cap off of the pupal case. The fly then can crawl out. That sac gets sucked back into the head, and then the eyes fuse shut, and it doesn't emerge anymore. Those flies emerge. They'll within the 10 to 14 day period, they'll find a mate, and then that after a period of development, that female fly will go seek a host to deposit her eggs. So the female, day 7 to 9 of their adult life, typically will be ready to lay eggs if she is mated, and she'll find open wounds and lay her eggs in it. Now, they don't lay one or two eggs, they'll lay 200 or 300 eggs at a time, which, as you imagine those things growing and getting larger and larger, they're going to consume more and more tissue. So you move from an organism that's out in the environment at large into one that is targeting animal wounds where these eggs are laid. Screwworm eggs are laid very distinctly. If you find an egg mass on a living animal, you can actually come pretty close to identifying them as screwworm if they're laid very methodically. They've been described to me as almost like shingles on a roof, and I think you can kind of see that on here of another picture later that'll demonstrate that more. Those eggs hatch after about 12 hours and they immediately crawl down into the living tissue where they start consuming it and creating all kinds of tissue damage that then that rotting tissue that they leave behind and all of their excrement causes the odors of a rotting carcass that draw in the other flies. So these larvae are in the wound. They tend not to be up on the surface. They will burrow down in and create pockets. And so that's why we talk about when you remove them, you need to make sure you get all the maggots out because a lot of the surface maggots you see are not screwworm. They're the other blowflies that came in afterwards. And you need to make sure we get the screwworms out of there. So after 5 to 7 days, if the animal hasn't died, the larvae will crawl out of the host and fall to the ground, and then they wiggle around and try and burrow into the soil if they're able to, and they turn into the pupal stage. And that pupal stage is like a chrysalis for a butterfly. It's where they change from the larval form to the adult form. That takes about a week, and then they pop that cap off and the life cycle continues. So this pupae are in the off host in the soil habitat. Adults are out in the environment, and then the eggs and larvae are on the wound or in the animal. All right, so mentioned eggs, you can see this female is laying these eggs very organized in how they're laid, whereas if you look at the bottom picture, the eggs are just very disorganized. These are the secondary screwworm on the bottom, and she's just running around laying eggs and doesn't really care how they are aligned. And again, they can deposit several hundred eggs at a time, especially if they're not disturbed. Right, 10 to 12 hours they hatch. You can certainly have multiple screwworm females laying eggs in a single wound, so it may be way more than 200 or 300 eggs at a time. And the more larvae, the more damage, the faster the death. Areas where they prefer to oviposit, navels are like candy to these flies. On newborn animals, that umbilicus area is the number one site where they will target. They also will target ears because of ear tags that cause abrasions or ticks that maybe are in the ears that leave an open wound. Of course, there's a number of things that humans do to our animals, such as castrating, dehorning, shearing, that can, in fact, create wounds that the fly will use. We also have other insects that create wounds, such as cattle grubs when they come up on the back of the animal and make their appearance. That may be an attractive site for these flies as well. And of course, tick bites. When ticks leave, they often have left a fair amount of damage. So why do we care? Because this thing will go into any mammal and occasionally birds, right? So, and us, right? So we're at the center of all of this. So cattle, horses, goats, sheep, pigs, dogs, cats, and wildlife. And wildlife is kind of the big difference between back then and now is what we're likely to see happen. Because when this fly was here previously, there was very little wildlife in southern Texas in particular than there is now with white-tailed deer and a lot of the exotics that we have. So I put this up just so you're warned. There are some pictures coming up that are pretty gruesome. And starting with this first one. So the larvae. So as I said, they feed for 5 to 7 days. They have to go through a living animal. If an animal dies while they're in the larval stage, those larvae will leave the host. And if they're large enough, if they're early third instars, they'll go through and complete pupation and just emerge probably as a smaller adult fly. So death of the animal doesn't necessarily mean death of the maggot. Now, if they're first instars, they're unlikely to survive, but they will not stay on that carcass and continue to feed, which has its own problems. with how we might dispose of, say, wildlife that are killed by screwworms. So I've mentioned before, it doesn't really care what it lays its eggs in. This is a picture of the back of the neck of a dog that has a pretty severe myiasis infestation. Now, all those maggots are not all screwworms in all likelihood. They're probably other blowflies that came in after the screwworm created the nasty wound on that animal. But as a veterinarian, you need to get every one of those maggots out of there and submit samples of those to be identified to make sure that if there are screwworms, we know about it. And some of the studies in Latin America have shown that 100% of newborns that are in areas with screwworms will have infested umbilicus. And a calf doesn't live very long if its umbilicus is infested because of where that location is. All right, so I mentioned the pupae drop to the ground, or larvae drop to the ground and pupate. They are in that stage for a week or so. It's important to understand that this is not a contagious disease. If you have one animal that has screwworm on it, yes, you need to check your other animals, but the fly larvae aren't going to move from one animal to another. Unless you had cuts on your hand and you left it there a while, they're not going to move from your cow onto you unless you stick your finger in the hole, you have a cut and some of the flies move over to you perhaps, but that's pretty unlikely, right? So since this fly escaped the quarantine zone about 4 years ago, there have been 1,000 human cases in Mexico and Central America and about 7 deaths. Now, most of the cases have been in Central America. They've been dealing with it longer, but most of the deaths have been in Mexico. And it kind of brings it home. There's probably reasons for why the humans are not getting medical attention, perhaps access to it, or other complicating factors that we can talk about after the presentation if you'd like. But it is real to humans, and it is a horribly painful way to die. So people always ask me, how far do the flies fly? Well, that's a difficult question to ask. Research has shown us that they can fly up to 12 and a half miles a day in order to find a suitable host. But I always ask people to put it in perspective, right? If you're hungry, so remember, this female fly has one job. She is to find a host to lay her eggs. That is her one and only job in perpetuating her species. But if she's flying along, flies out of her wooded area and finds a cow and drops her eggs on that cow, she's not going to go, all right, great, I'm going to keep flying for 10 and a half more miles. I need to get my 12 and a half mile step cred in today. That's not what their motivation is. And so if there is a lack of hosts, yes, they can move 12 miles, but it's like you driving down the interstate and you get hungry and you're hungry for, say, McDonald's. Are you going to pass 7 McDonald's so that you can go 12 miles to make sure that you went your full distance for the day? No, you're going to stop at the first McDonald's. It's on the correct side of the highway that looks like you can get your food you want, right? So historically, when this fly has moved around the U.S. over great distances, so from, say, Texas up to Nebraska in the summer, it's been humans putting infested animals onto trailers and driving them hundreds of miles without either hopefully not knowing that those animals were infested. Once the animals up in the Midwest, maggots continue developing, fall out, produce adult flies, and now you have infestations in Nebraska. So we need to keep that in mind with how we do a response. All right, I want to kind of jump through some of the history of screwworms and the eradication. So the image up here is one of the key deer from the Florida outbreak in 2016. And that outbreak really was centered on the bucks that were shedding velvet at the time. And so the access point for a lot of the screwworms were up on the head, which was not a good area if you're a key deer to have a screwworm infestation. This is a native species to the Americas. Even though it's called new, it is not new. It is not an invasive species. We have writings from when the conquistadors arrived describing some of the maggot infestations on some of the folks they had with them. And so we know they encountered them. And, this fly is native to our hemisphere. And then when the conquistadors made their ventures up into the southwestern US, it was already present. In the 1858, a scientist named Cochliomyia gave the species name of Cochliomyia hominivorax, and that was done with specimens he collected in French Guiana and Northeast South America. Now, if you look in the Wayback Literature, it was a different genus than Cochliomyia. It's been renamed since, but the hominivorax is original. So you see on the map, you have all the red areas in the south, and then a big kind of Georgia's disappeared underneath a red splotch. That's because in 1933, Georgia suddenly had an outbreak of screwworm and it moved into Florida. And those are thought to have been cattle that Texas had a drought in 1933. And we ship animals out when we don't have grass to feed them. Some of those animals that had infestations were shipped to Georgia, and suddenly the southeastern US had a screwworm infestation. But if you look at the eastern edge of this red area, the large red area, that's the Mississippi River, which was a biological barrier for the fly to get into the eastern side of the river. But again, humans helped move them across that river. In the early 30s, two scientists actually were able to clarify that there were two screwworms present in the Americas, that the New World screwworm and the one we've been calling the secondary screwworm. That's absolutely critical because in entomology, you can't solve a pest problem unless you know what the pest is. And by identifying that we actually were dealing with two different flies with very different biologies from each other allowed us to make great strides on eliminating the New World screwworm. So there are two scientists here, Edward Knipling and Raymond Bushland. They're here. This is a picture taken in Kerrville, Texas. That laboratory is actually named after these two scientists. And on the right are the young and older pictures of Dr. Knipling. Dr. Knipling spent his entire career in the Ag Research Service. He ended up leading the organization later in life. And I'm proud to say he is a graduate of the Department of Entomology at Texas A&M University. He is the one who came up with area-wide pest control that we now call Sterile Insect Technique. And it was him working with Bushland and some others that refined that process. And we're going to kind of talk a bit about that. So Melvin and Bushland created a way to mass produce these flies, right? So part of a sterile insect technique is you have to produce many millions of these flies. Well, this is a fly that ate living organisms. And so sacrificing living organisms in order to mass produce them was not ethical and not something anyone wanted to do. But they were able to use horse meat and some other things and essentially get the screwworm to develop on non-living hosts, which was a huge step forward. And after they did that, they had a mechanism to raise lots and lots of flies that we still do today. Knipling and Bushland got together and came up with this concept of the sterile insect technique. They did that by understanding the fly's biology. The female New World screwworm will mate only one time. And the concept is if you can get her to mate with a sterile male, she becomes reproductively dead. She'll still lay eggs, just like chickens lay eggs that we buy in the store. Those are non-fertilized eggs. They'll keep laying eggs, but none of those eggs will ever hatch. So we had to find a way to sterilize them. So these guys were working pretty hard on this in the 30s. And then World War II came about. They all went into deployments, returned from the war, went back to work, and started working on the screwworm problem again. Dr. Knipling grew up in Port Lavaca, Texas, which is kind of north of Corpus Christi on the Gulf Coast. And he, in his writings, he writes about how horrible the screwworm was for him as a child. And he was able to dedicate his career to getting rid of this fly. So Bushland and Hopkins started working on how do we sterilize the flies, which is its own problem because you have to be able to sterilize them without affecting their behavior or their acceptability to the wild flies. So if you sterilize the flies and put them out there, and the wild female flies encounter a sterilized male fly, and they don't recognize it as a male screwworm, they're not going to mate with it, and your whole program doesn't work. So they looked at, during World War II, we had the atomic energy discovery, and they looked at X-ray and chemicals, and eventually moved on to gamma irradiation to create the sterilized insects that were used in the program. So then they had their ability to grow the flies, to sterilize them. They identified that they thought they could disperse the flies over large geographic areas using small aircraft. They were ready to go, and so they needed to demonstrate that it worked. The first place that they went to was a small island off of the coast of Florida, south of Tampa, called Sanibel Island. Sanibel Island is just a few miles long. It's about 3 miles maybe off the coast of Florida. And they went and released sterile flies and very rapidly eradicated the fly from the island. The problem is, after a few months, the flies essentially moved back from the mainland back onto the island and recolonized it. There's a big bridge that they likely followed to get back over there, but they had demonstrated to themselves that they could do it. So then they decided that doing this on an island in the Caribbean that had screwworms was a better place to show we can eliminate them and demonstrate that they truly are eliminated, not that we just couldn't catch them anymore. So they went to Curacao. So Curacao is one of the ABC islands off of Venezuela. So Aruba, Bonaire, and Curacao. Curacao is more of a deserty island, and they went down there. It's about 30 miles from any other island, so very isolated. And they very rapidly eradicated screwworm from Curacao using SIT. That was the proof they needed to get the leverage to then look at how do we eradicate it on mainland US. So as this was going on, Knipling was formulating more of his conceptual ideas with sterile insects, such as how many generations it would take, how many flies would you need to release, some of the math behind it. And I will say that almost everything he predicted or that he suggested would be needed did in fact play out. So at that time, the screwworm production plant was in Sebring, Florida, which is just north of the Everglades, and they used that. And so in 57, they started the eradication program in the southeast, and they're not dummies. They waited until winter when the fly died back down to the Orlando area, and they only had to really eradicate the southern half of Florida, and they did that in about 2 or 3 years. So back over here in Texas, our ranchers were watching with great envy as to what was happening and the successes that they were having in Florida. And they formed a foundation called the Southwest Animal Health Research Foundation and fundraised almost $2 million in the early 1960s to build a screwworm production plant in Mission, Texas, which is way down here in South Texas and near Edinburgh. They essentially tell me that they went door to door asking the ranchers to contribute for this. They built that facility in about 18 months, is what they're telling me, and handed that plant over to APHIS to start producing sterile flies in Texas. So in 62, they started what they called the Southwestern U.S. eradication program. Very rapidly had success. They produced millions and millions of flies, and their target release rate was 400 male and females per square mile per week, right? Remembering that when they're producing these flies in the culture, that they are producing males and females, eradicating everybody and releasing males and females. It didn't matter that they were releasing females because they're sterile. But this will play a part in later in how we hope we can move this forward. So they had rapid success in the Southwest, and they considered Texas to be eradicated after 2 years, along with New Mexico, but we kept having reinfestations. So they started doing releases into northern Mexico during the winter to try and suppress the population in northern Mexico that was thought to be the source of the Texas reinfestations every year. As that happened, we had a couple of incidents happen over the years. And so this is a graph of the number of reported screwworm cases in Texas between 1962 and 1983. And recognize that in 62, they started the SIT program. They dropped from over 1 million cases a year in 1961 and earlier to 50,000 cases in Texas. That is absolutely remarkable. The year after, they were down to 5,000 cases. And this is where Nippling had predicted these numbers. And they still had cases every year, but then in 1972, they had an outbreak from a warm winter of 90,000 cases in Texas. At that point, the ranchers recognized and USDA recognized that as long as the fly was in northern Mexico, it was going to be a constant threat to the US. And so they started working with Mexico to develop a binational eradication program. Of course, they started releases and knocked it back down. We had another little mini outbreak in 76. And then as we pushed the fly further south in Mexico, we cleaned up all those cases in Texas. And by 1983 was the first year we had 0 cases reported in Texas, and they had not had another case in Texas since. So that was considered the full real eradication date was 1983. So this just summarizes some of that. So in 78, they built a plant down in southern Mexico, down in, I think it was in Chiapas, and started producing flies. And where I think we made a mistake was that in 1983, we closed our production plant in Mission. And the reason that they closed these plants are that it costs a lot of money to fly these flies all the way down to Central America, or now we're flying them from Panama to Texas, and having more local production allows you to get those flies out there healthier. They'll live longer and maybe do more work for you before they die. So Mexico went through its eradication program. It wasn't considered eradicated until 1991. So it took them almost 30 years to eradicate Mexico if we consider that they started in the early 70s. Right, we have introductions of screwworm in this country, despite what sometimes the media says. So there are I'll call it frequent, several times a year. In some cases, we have animals show up at some of our airports that have screwworms if they're coming from Latin American countries that have screwworm, or some of the Caribbean islands. And so in 78, they found a dog at the Miami airport that was infested. They removed those maggots, released sterile flies, and never had an episode. Later that July, there was a dog that came back with some military personnel to Panama City out here on the Panhandle, and they rapidly removed those and did not have an outbreak. So in the late 70s, they moved the program. They started making arrangements with the various Central American countries. And the idea initially, when we worked with Mexico, was to eradicate it down to the Isthmus of Tehuantepec, which is the narrowest area of Mexico, thinking that if we have it in a narrow area, we can really do an easier job, less expensive job of containment. Well, at this same time, the Central American countries were very interested in getting rid of screwworm, and they decided to use the Isthmus of Panama, the narrowest area in all of the Americas continents, as the stopping point. And so in the early 90s, Panama and the US formed a group called COPEG, and the acronym is better translated into Spanish, but it essentially is a cooperative program with Panama and the US to support this production plant that you see on the upper right. That's where all the screwworms in the world are produced right now, and have been for the last 20 some years. So this building is larger than the biggest Walmart you may have ever been in. And the idea is the plant sits on the east side of the Panama Canal, the South American side, and this area is called the Darién Gap. And they decided that the Darién Gap would provide a biological barrier to screwworm movement. And that worked for about 20 years because this was an impenetrable jungle. Well, more recently, there have been some clear cutting or tree removal to facilitate livestock production in the Darien Gap, which now provides very susceptible hosts for the screwworms. And one of the reasons we think it came back was that it built up populations in the Darien Gap much higher than they had historically been. And then with some complications from COVID and perhaps human movement through there, that we happen to have the stars align in a bad way for the screwworm to escape the quarantine and see what we're suffering now. All right. So once the Copeg plant became operational, the plant they had built in Nicaragua closed, and we had one production area in the world for this. And that's really not good for a fly this important. And then we jumped to 2016. We had no screwworm issues until 2016 when Florida Keys had their outbreak. So the Florida Keys are a string of islands off the tip of Florida. Most all you ought to know, Key West is the last island, but there's one highway, US 1, that runs the length of all the islands. So one way on and off the islands by car. Big Pine Key sits out toward the end of that. And what happened essentially was a dog was presented to a veterinarian sometime between April and June with maggots on it. The veterinarian removed the maggots, should have submitted those to NVSL for identification, but didn't, but did save the maggots, thankfully, and didn't report it. And so APHIS was unaware that there was a screwworm problem, or screwworm had made an appearance on Big Pine Key. Later that fall, so the key deer is a small white-tailed deer. It's a subspecies of white-tailed deer that's considered an endangered species. And it only lives on a handful of islands that are in and around Big Pine Key. Many of the deer on the island are very habituated to humans. The tourists like to feed them. People name them. They have feeders in their yards. And in late August, early September, the deer start to shed the velvet on the antlers, and that opened some wounds. And the screwworms that had now been on the island for a few months, larger populations, started attacking the key deer. And it was really the local humans that recognized something was wrong, alerted their officials, who then notified APHIS, confirmed that it was screwworm, and rapidly mobilized to get down there. And so this graph on the bottom shows the distribution of flies that APHIS caught. So they started releasing sterile flies in early October, and initially the red are the wild flies, and very rapidly you see the wild flies. The red and orange are wild flies, and they rapidly eliminated screwworms on the island through sterile insect technique and through management of the infected key deer. There is one fly that was found in early January, and if I remember correctly, that fly was actually found up in peninsular Florida. That was a one-off. They never found it again up there, and it was an infested dog that they found some pupae in the area when that dog had been identified as having a wound. But one of the things we did was containment of the organism by having a mandatory quarantine and inspection area here on Marathon Island. Every animal that left the Keys had to be inspected and had to, I think if I remember right, they had to have a health inspection certificate. And that helped to keep anyone from moving a sick animal onto the mainland of Florida. But in the end, APHIS deployed 539 people, or 350 people. Some of them had to go multiple times. They released 188 million sterile flies. They did ground releases in this case because the area was small. They medicated deer. So they were feeding doramectin on bread to deer that would hand feed and then setting it out in the island to try and treat those deer with a medication that might kill the maggots. They really have no idea whether that worked or not. And then they were properly composting deer carcasses as if they had to euthanize animals. In the end, the overall cost was over $10 million for what we would call a fairly small infestation. And it was considered re-eradicated in March of 17. So it took about 8 months for them to fully eliminate that fly infestation. All right, the current outbreak, and I need to speed up because we're getting close on time. So in 2022, Panama regularly used to have screwworms on the western side of the country, and they averaged about 25 cases a year that they dealt with. And they normally are releasing sterile flies around that plant in that area just to make sure nothing moves out of it. But they jumped from 25 to 6,500 cases. They started cranking up production of the sterile flies, but it was essentially gotten ahead of them. The next year, it showed up in Costa Rica, then it moved through all of the countries. And where we really got involved was in November of 24. It moved from Guatemala into Mexico with a couple of cases. And that's when we really started to mobilize. It moved into multiple states in southern Mexico. And later, last May, April, May, there were some cases in Veracruz and Oaxaca. And APHIS went in and kind of snuffed those out pretty quickly. The U.S. had closed the border to Mexican cattle when it first showed up in Mexico. So normally we import 1 million to 2 million head of cattle from Mexico every year. Most of those animals go to our feedlots. They shut the border down in January. They reopened it a month or a couple months later. And then probably more to do with politics and getting Mexico to comply with what APHIS wanted them to do to manage the infestation, the USDA closed the border to cattle again. So there are 5 places along the US-Mexico border where cattle are imported, 1 in Arizona, 1 in New Mexico, and 3 in Texas. And it has been devastating to our commercial feedlot industry that they don't have these animals, and we're starting to see some repercussions. And this is on top of the U.S. having one of our lowest inventories of total cattle that we have had in, I think it's 70 years. So that's why you're seeing beef prices so high. All right, so they had made a plan after everything was very good for several months that in July they did reopen the border in Douglas, Arizona. It was considered the safest area with the least hospitable environment. And they had that border open, had it open for 2 days. There were various inspections that had to be done. And then a case showed up 370 miles from the border, and USDA closed all the importation border sites again to cattle, bison, horses, and essentially all livestock. And they have never reopened. So they have been closed for really a year or more in all practical purposes. So what's happening when we look at Central America? These are old data. They're not really updating anymore because it's gotten so out of hand. But in 2 years, Central America had reported 50,000 cases across all of the countries. But you do see on the left, it's cattle that seem to be taking the most hits. But keep in mind that all sampling is biased, and humans are able to look at their dogs, their cows, their pigs, those animals much more easily than they are wildlife. And so you do see some wildlife at the bottom from Central America that have had infestations, but it really has been reported as a livestock issue. The current outbreak, and this map is back from January 18th. So Mexico is considered heavily infested in the southern states, moderately infested in some of these others. And the numbers in purple are the number of cases. This outline here is really where they were doing the New World Screwworm releases. They're now moving that line up to encompass southern Texas. But when we look at as of last August, Panama itself had over 50,000 cases with 162 humans. And you can read the other numbers here. As of last Thursday, and the numbers for Mexico get updated every Thursday, and you can see those on screwworm.gov. You can see the new numbers. Cumulative Mexico since November of 24 has had 15,000 cases, and it was 8,800 back in October. So it's been very consistent in how many. Then they have 710 active cases that they consider they're actively working, and all the others have been resolved. Again, just like we saw in Central America, our livestock species and dogs are the most affected or reported. Mexico's had 117 human cases and more than 7 deaths. And then in July last year, we had that's that Veracruz. So it was actually, I think, down in this area is where that infestation had happened that closed the borders. Nuevo Leon, we had infestations up and above and below Monterrey at a couple of feedlots from animals that had been moved from southern Mexico. But in those cases, those animals had been treated properly and they intercepted them through the procedures that they have to follow. The one that's most concerning is the day after Christmas in Tamaulipas, 197 miles from the border, we had a 6-day-old calf that was infested. Calves don't generally move, and that animal had no movement history, which means there is an adult fly in that area that landed on that calf and deposited her eggs. So that's local transmission, and that is very, very concerning. And since then, we've seen lots of cases in Tamaulipas. So as of last week, since January 1st, there have been 136 cases within 400 miles of the US border. And that's a number that you can count up every week. They'll post them and what states they're in and what animal they were in. And that really has upped the game. They moved the sterile insect releases from down here to up in this area to try and keep it away from Texas. And they recently just announced they're going to start releasing even further north. All right, so Mexico and APHIS had agreed to a protocol for moving animals out of infested areas, and that entailed any animal being moved had to receive an ivermectin application 3 days before it was moved, and they had to conduct an inspection before it left the area and then when it offloaded to its destination. And in the case of the Monterey cases, both cases were found when they offloaded those animals. And that's where we have these in Nuevo Leon. All right. We did have 2 cases in the US last summer. There was a human case in Maryland from a traveler who had gone to El Salvador, came back, realized they had something wrong, went to the doctor, and they had screwworms on them. These don't generally get reported because CDC is not in the business of reporting because of protecting privacy of patients. But oftentimes we hear about it because patients will go on social media or tell people, and those people tell people, and it gets in the news. Here a few weeks ago, there was a horse imported to the Miami area from Argentina, and it had screwworms on it. So nothing related to the Mexico problem we have now. This was a traditional moved from Latin America and had screwworms. They caught the screwworms. They released flies, I believe, but they don't consider it to be a risk for an establishment. And then I mentioned this one that's very, very concerning of the local transmission in Tamaulipas. So how do we survey for screwworms? So this is, if you go to Home Depot, you can buy black drainage pipe. USDA uses this trap now because it's proven to be pretty effective. They cut them into 12-inch or 15-inch pieces, and then they cover them with a sticky substance called tangle trap, which is a really nasty stuff that we use in entomology to catch insects. They attach 2 of these, which is just clear Tygon tubing, stuff it with cotton. And this gentleman is taking the Swarm Lure 4 product with a syringe and injecting it into the cotton wick. It gives off an odor that the only thing I can describe it as is if you've ever smelled a deer that has been gut shot or you're cleaning a deer and you cut open the rumen and you get that odor along with blood. It's truly offensive, but it is attractive to not only screwworms, but all other blowflies. You can see one while we were setting these up had already attracted a blowfly. So people go out every week and either pull these individual flies off, or if there are lots of them, they'll scrape them off and then they try and identify them, which can in itself be a problem when they're coming off a sticky trap and you're looking at little tiny hairs on their face. So we often use molecular backup to do confirmation. So this is the red areas where, and it's just my overlay of a guesstimate as to where they're going to do the releases. I haven't seen the map, but it's likely these 50 miles into Texas. Um, and it really is to try and make sure that we don't have that fly show up in Texas. So they have been monitoring along the Rio Grande from Del Rio all the way down to the border. 101 of those traps have been in place for about a year and have not yet caught a screwworm, but they have to move those traps up, probably up into this area, because they don't want the traps in the area where they're releasing the screwworm, sterile screwworms, because they'll be just as attracted to the trap as the wild ones. All right, why do we care besides the pain and suffering of our animals? These are some numbers that A&M came up with about a year ago, strictly based on inflation and what we know the costs were back in 76. They had updated it to 2016 when Florida had the fly, and then we re-updated them last year. And so the dark blue is losses to the Texas producers, which just for cattle is estimated to be over 700 million. But when you look at the Texas economy, it balloons up to $1.8 billion. And this is just cattle, right? So when we add in wildlife, the wildlife industry is worth more than $10 billion to the Texas economy through hunting fees and others, as well as property values. So our wildlife are at great risk because we have no way of catching them, of treating them, and the native wildlife are very much protected by hunting rules and regulations that our parks and wildlife are actually looking at ways to help with that. Right? If I see a deer, I have binoculars on it, it obviously has myiasis. Can I shoot that deer and make sure that those flies, maggots don't become adults? And that's actually on the board right now, but you have to go through a process to do that because we don't just want people poaching deer all the time. Right. So what falls into the cost of production? So obviously animal deaths, decreased production, veterinary care is a big one. Probably the biggest is needing to have people who can put eyes on your animals all the time. The ranchers tell me that the number of cowboys that they are able to employ with given today's costs is minuscule compared to what they had been. And we are talking cattle on vast, vast tens of thousands of acres trying to find animals, right? So what we talk about is, as a rancher, think about what you can do to plan ahead, right? What activities do you do that cause wounds to your animals that might facilitate a screwworm attack, right? Maintain your facility, your fences. Do you have places that animals have cut themselves in the past because there's a pipe sticking up or something? Aggressive dogs, animal handling, sire choices, right? So cattle folks, like, you can pick a bull that sires smaller calves so that the female isn't as damaged when she's giving birth, because the screwworms can attack the females after they've given birth as well. One of the things that we're looking at is pre-treatment with systemic insecticides such as doramectin or ivermectin. And like, so if you're going to do a castration event or a dehorning, treat those animals when you do that so that if a screwworm lays eggs on your cow, those larvae hatch, the ivermectin's already there and it kills them when they're little, and you've protected them. That wound has time to heal before that chemical leaves the body. Do you have the ability to capture and hold your animals to treat them, right? There's no way you're going to lasso a 2,500-pound bull out in the middle of a pasture to treat it. So how are you going to handle that if that animal has an infestation? What are the behaviors of your animals? What level of veterinary care do you have? We have a lot of ranches that don't have veterinary care because there's not a veterinarian in their county that does large animals. That's a problem because some of our treatments require a veterinary client relationship, right? And any delay in treatment can result in fatality. So this fly grows very rapidly. And if you don't get to it quickly, you may come back in 2 days and your animal is dead, right? And then it of course has economic costs. So management of screwworm is integrated pest management. Anything we can do to reduce the population of the fly in the environment makes those sterile males all the more effective because they have fewer females to try and find. So we talk about inspection and surveillance. Put eyes on your animals every 2 or 3 days. Take a look at them, watch their behavior. Can you put them in places where you have the ability to work them? Um, we talk about not just using something like an ivermectin continuously because you're going to select for resistance, not necessarily to screwworm, but to all the other parasites that are attacking your animals, right? We don't have a lot of chemical options, although the options we have are way better than we had in the 1960s. But always remind people eradication is the goal. We are not trying to manage this fly. We are trying to eliminate this fly. So that includes surveying for adults. We need to know where they're at so that we can adjust our sterile insect releases. We need to monitor for the larvae, which requires those animal in examinations. We need to conduct inspections and limit animal movement in areas where we know screwworm is circulating. And of course, work with APHIS to get those sterile insects released into the area. So I talk about rules and regulations on pesticides. You can't just use a pesticide because you know it'll work. There are labels that are tied to these pesticides that are absolutely critical so that we don't compromise our food safety for the sake of keeping an animal alive. Because if that animal is tested at a slaughter plant and found to have residues, they will condemn that animal and they will back trace it to your ranch. Um, right. So harvested wildlife is a problem, right? So a lot of folks want to feed white-tailed deer ivermectin to save these beautiful animals. And I'm all for saving those animals, but ivermectin in cattle has a 35-day withdrawal period. South Texas, when are you going to stop using ivermectin? The deer season starts in August and runs through February in some places. Screwworms are active during a lot of that time. So maybe there are times of the year that it would be appropriate to use them, but we also don't necessarily have um, residues set for things like wildlife because it's quite expensive to do those studies, and the use for the company to recover those costs is quite small. So this one's kind of cut off, but people always talk to me about what we used to use this. Why can't we use that? The reason is in the far right column. All of these pesticides that we had in the 60s and 70s have been canceled. There's only a couple left, and but a lot of them that folks remember using, really you can't use anymore. And a lot of them are not even produced anymore and a lot of them are old organophosphates that we'd rather not have back anyway. So we talked to our friends and colleagues in Latin America. They've been dealing with this fly forever, particularly South America. And they talk about well-managed operations have fewer issues, right? If your animals are getting cut less, they're going to have fewer flies. So putting in insecticide ear tags that can help protect from tick bites. So you have fewer ticks, you have fewer places for screwworms to lay their eggs. Look at your animals regularly. Conduct maintenance on your things. We've talked about some of these. Treat wounds properly. So if you know your cow is about to give birth to the calf, maybe stick around once that calf drops, treat that umbilicus with a product. We just had last week, ivermectin became a registered use, and you can use it on a newborn calf. And so that's one of our newer recommendations is if you are in an area where screwworm is active, it would be a very advisable thing to treat that calf with an ivermectin injectable because you're now going to give it a week or 2 or 3 weeks of protection while that umbilicus heals and your calf doesn't die. There are some other products, but there's some that have calf restrictions. When we talk about registering products, Just very quickly, you have FDA, which does drugs, so things that are absorbed and systemically distributed, and EPA is more the surface. There are some important considerations for veterinarians who are permitted to use extra label use. So if a say a product is registered for a sheep but screwworm isn't on it, a veterinarian has the option of treating that animal for screwworm if that product is labeled. With an EPA product, not even a veterinarian can do that. The label is the law, and you cannot use an extra label. So unless screwworm is on the label, you can't use that EPA product. Unless cow is on the label, you can't use that product. And so EPA has been more challenging to get new products listed, but we do have some, and we're working on some other ones. So we work with FDA and EPA to try and identify products that would be useful. So the SIT program. So this is some pictures from COPEG. You can type COPEG into Google and go watch these videos. As I mentioned, there's one plant in Panama that produces them. They produce about 110 million flies a week, and half of those are males. They're renovating a plant in Mexico that will produce 100 million flies, hopefully by the end of this summer, which will greatly help our efforts. And they're building a new production plant in Edinburgh, Texas at Moore Air Base that, when it's fully online in a couple of years, will produce 300 million flies. But by the end of next summer, summer of 27, they hope to produce 100 million flies. We are going to need all 500 million flies if we're going to push this fly back to that containment zone. And this is going to take decades, right? The larvae are reared on artificial diets, so they use like a dried blood product from slaughterhouses and some other things to rear them. So no animals are directly used. And they do this on an industrial scale, 7 days a week, 24 hours a day, 365. And so once they harvest these larvae in the upper picture, they allow them to turn into pupae. And then at a certain age, like I think it's 5.5 days, they put them in about 2-gallon containers, stick them in a chamber that has cobalt-19 in it, emitting radiation, and they put them in there for a few minutes, take them out, and now those flies are sterilized. They package those pupae up and ship them up to the site where they're going to disperse them. So now they're going to send a lot of those to Edinburgh because we just opened the dispersal facility there. They let the flies eclose, put them in these boxes. I'll have some better pictures here in a second, chill them down to about 60 degrees and get them loaded on the airplanes. When they do these distributions, they fly very specific patterns, GPS directed. So here they're harvesting pupae. These are aerial or ground-based, I should say, ground-based dispersal sites. So the pupae are in here and they just emerge as adults and fly out. So we tend to do that in more local areas where you want to there's screwworm pupae or adults in that area, and you want to just overwhelm that particular area. They use that approach. So the idea is you want to make sure that there are so many of your sterile males in an area that a wild female has a phenomenally mathematical chance of encountering your sterile male that she chooses to mate with, and then she's done mating forever, and she then becomes essentially reproductively dead. So when we kind of historic versus current, so some of you may remember finding the little boxes on the ground that had sterile flies. That's how they did it in the past. They don't do that anymore. They would package up the pupae in the little boxes and drop them out of the airplanes, fall to the ground, the pupae would emerge, or the adults would emerge and then fly off. Now they've gone to this packaging them in these big boxes. The boxes sit on top of this auger system in the plane, and then there are baffles that open underneath that the adult flies fall into, and a computer system runs the augering so that over an area where perhaps they know they have a number of cases and suspect adult flies, and the habitat is good, they may release more flies there than areas that are less suspect or less supportive of the fly, so making the most of those flies. So this is, those of you that do mosquito control may recognize this sort of a thing. When you do mosquito control, you fly a grid pattern trying to get your insecticide to drift over an area. Well, in this case, we're dropping flies so that they fall down over an area, and it's all based on some computer technology with GIS and where the plane's at and the number of cases. And so it's very scientific in what they're doing, very good at that. So I'm getting close here. I know we're running out of time, but um, what we want is if you encounter screwworm or think you do, please report it. Any maggot infestation in a live animal has to be reported. It is federal law. But you can watch your animals for signs of screwworm. Are they irritated, head shaking? Do you smell decay? Um, do you have one animal that's hanging away from all the other animals because it just doesn't feel well? Think of yourself, right? When you're sick, you don't want to hang out with other people. And so, do you have one that's acting funny? Then pay a little more attention to that one. So USDA has really stepped up in this fight, and as I said, they provided $21 million to renovate a fruit fly plant in Mexico that should come online at the end of this summer to produce 100 million flies. This last June, they announced creating the dispersal facility, so moving the flies from Panama to Texas, repackaging them, and getting them into the small aircraft. That just opened on Monday. They did the ribbon cutting, and so it's ready to go. And and then the production facility is being built in South Texas next to where the dispersal facility is, and they expect to produce 100 million flies by the middle of next summer or end of next summer, and then 300 million flies within a couple of years of that. Texas Animal Health Commission has developed a response plan and protocols working with APHIS Veterinary Service, which will be the group that does the domestic eradication. And then USDA announced, I'm not going to go through these, but USDA announced the five areas where they want to focus. So innovate to eradication, keep it from spreading from Mexico, et cetera. All right, response plan. So New World Screwworm is a foreign animal disease pest, which means it falls directly and only under USDA APHIS Veterinary Services control for eradication. They partner with state ag organizations. So in Texas, it's Animal Health Commission, largely in Parks and Wildlife. They have some involvement, I think, with Department of Agriculture. They have developed the playbook, which is really the details of what a response would look like, and have vetted that with experts that have gone through some of these scenarios with them. States themselves are developing their own response plans, and Texas is no different. Texas has the plan to maintain the continuity of business, right? We want businesses to stay in business. If you need to ship calves, we're going to try and find a way that you can do that while we protect the rest of agriculture in Texas. And the veterinarians, state veterinarians across the Southwest have been getting together to make sure that states will accept, say, Texas cattle. If we have an infestation in one part, they're not going to shut the entire state of Texas down. So tribal groups in some parts of the country will need to interact with their state ag departments. And this is quite different from the Florida outbreak in that we have had a year to get ready for the arrival of this fly. In Florida, it showed up and it was very fast response. We have had time and we really have done a great job in getting ready for this. Okay, so SIT is how we're going to re-eradicate this pest. Now, there are some things we can do to help that, but SIT is the path to re-eradication. We hope to, well, we also want people to recognize when we eradicated this the last time, we had 500 to 700 million flies per week, and it took decades to eradicate. The fly, when it moved from Panama to southern Mexico, it did that in about 2 years, but it took us 17 years to eradicate it from that area down to the Darién Gap. We are in this for a while, folks, and we need to pay attention to this. And there is hope with the genetically modified sterile fly. They have that. They're testing it out that some of these new plants may be able to produce that, which would essentially, they're male-only strains, which would allow us to double our production because they're all males. All right. I don't think I have time to go through all
Yeah,
of these.
we're coming up pretty close on time.
Okay. Let me just pop through. So there are some products that are out there, um, and you can go to the USDA APHIS site and see these. A couple that are really important is this Fluralaner product, Exalt, is conditional approval. Dectomax and Exalt have to use a veterinarian client privilege to use. Exalt is going to be great, but you can't use it on calves under 2 months of age. Ivermectin you can, so that's a very good addition. Um, and there are some products for cats and dogs. So this Credello product you can use, and these are systemic, so they will provide some lasting control. They're not always that cheap. Um, what I tell ranchers is develop a disaster management plan, right? They have them for hurricanes and other things, and there are steps you can take. We've gone through that in this presentation. Contact your authorities. So in Texas, every county has an extension agent. If you can remember nothing else, at least remember contact your extension agent. They are all trained in what to do. Texas Animal Health Commission, Texas Parks and Wildlife, if it's a wildlife animal, and every state has these sorts of things. Um, and then there are different states have different reporting structures, but essentially see something, say something. This is a winged animal. It will establish if you don't. And I'm just going to leave this up and we can take some questions if folks want to take a picture of this. But here are some websites that I gathered that have a lot of useful information on them. So you can go to this top one is a screwworm.org, and it has the most up to date. And if Screwworm shows up in the US, that's the website where they'll announce it. And with that, I'll stop. And if there are any questions and we have time, I'm happy to do that.
Thank you so much, Dr. Kaufman. I think we might have time for one or two quick questions, but I was captivated. I probably should have jumped in and said, "All right, let's go over the questions." But you really set the kind of the pace for the rest of the webinar series, and that was super comprehensive and very useful. So thank you so much for that.
You bet.
Um, let's see. I see two kind of short questions. Let's go with these. The first is, has the SIT or sterile insect technique been successfully used to control other agricultural pests or arthropod vectors relevant to human health? And they mentioned Anopheles gambiae.
So I believe that they have used it to an extent, slightly different way of using it for Aedes aegypti in a few places. Um, I don't think have used it for Anopheles gambiae at this point. There've been talk about it. So mosquito sterile insect technique is fairly new compared to some of the others. There's a lot of fruit fly sterile insect technique that happens. Um, so mex fly, med fly, and some of those are actively being targeted with sterile insect technique. But this is the classic. This is the one that we had it nailed, and I never ever thought I'd be working on this pest, and here I find myself in the middle of it. But no, good question.
And then there's a few clarifications in the Q&A, but here's I think a good one. How many times can a sterile male mate, or for how many days is it active?
Right. So the flies that are sterilized, I don't think there's any limit on the number of times it can mate. It's probably more opportunistic that it needs, whether it's available to mate or not, the females. Um, but these flies, their DNA has been trashed, and so those even the non-sex cells are not able to reproduce. So it's really a dead fly walking, right? So once something critical in that fly goes down, that fly's probably dying. And I think what I've heard, and if anyone's online wants to correct me, I think that they expect that the most of the sterile flies live 7 to 10 days.
All right. Well, thank you very much. I see we are a little bit past time actually, so I'm going to go ahead and wrap this up. Maybe what we can do is we can harvest the rest of the questions, and I can send you an email, Dr. Kaufman, and maybe we can answer those questions and send them out in our newsletter.
Certainly.
All right. I want to thank you again, Dr. Kaufman, for an excellent presentation, and thank you to everyone who joined us today and contributed to the discussion. So this was the first presentation in our series, The New World Screwworm, Past Lessons, Present Programs, and Future Frontiers. We'll hope that you'll join us again for upcoming sessions as we explore additional perspectives on screwworm history, current preparedness efforts, and emerging tools and approaches for the future. Next week, we actually don't have a webinar, but we will be back on the 25th to hear from Denise Benil from Animal Plant and Health Inspection Service within Veterinary Services, and she will share how the Department of Agriculture is preparing for the New World Screwworm Northern Advancement. Additionally, we invite you to visit the Gene Convene Virtual Institute if you're interested in learning more about genetic biocontrol, and to subscribe to our free newsletter to stay informed about future events and activities, and maybe see some of those other questions answered. But thank you again for your time and participation, and we look forward to seeing you at the next webinar. Hope you have a great day.